Hello, we're glad you've joined us for this live webinar, CASP Genotyping, Better by Design. I am Judy O'Rourke of LabRoots, and I'll be moderating this session. Today's educational web seminar is presented by LabRoots, the leading scientific social networking website and provider of virtual events and webinars advancing scientific collaboration and learning. It's brought to you by LGC. LGC is a global leader in the laboratory services, measurement standards, reference materials, genomics, and proficiency testing marketplaces. LGC's brand is born from precision in what it provides for its customers and in how LGC leads the way in advancement in many areas of science. To learn more, visit www.lgcgroup.com/genomics. Let's get started. You can pose questions to the speaker during the presentation while they're fresh in your mind. To do so, simply type them into the drop-down box located on the far left of your screen labeled Ask a Question and click on the Send button. Questions will be answered after the presentation. To enlarge the slide window, click on the arrows at the top right-hand corner of the presentation window. If you experience technical problems seeing or hearing the presentation, just click on the Support tab found at the top right of the presentation window, or report your problem by typing it into the Answer a Question box located on the far left of your screen. This is an educational webinar and thus offers free continuing education credits. Please click on the Continuing Education Credits tab located at the top right of the presentation window and follow the process of obtaining your credits. I now present today's speaker, Jonathan Curry, PhD, a senior scientist in scientific support at LGC. Dr. Curry studied molecular biology at Birkbeck College while working at GR Micro. The company was acquired by Quotient Bioresearch, which is now the LGC Group's Fordham facility. He received a PhD in microbiology from University College London. Dr. Curry used a wide variety of molecular biology techniques and sequencing during his PhD studies. He joined K Bioscience/LGC Genomics in 2013 to help build a technical support team and help customers get optimal results. One of his first roles was to implement sequence analysis into MagicBox, LGC's assay design software for automated whole genome primer analysis for all species. Dr. Curry will now begin his presentation.
Yes, thanks very much for the introduction. So welcome to Better by Design. We're going to talk about simple tools for effective CASP assay design. So what I'm going to go through is a little bit of who are we, and then how does CASP work, very briefly, just so we get an introduction. And then we're going to look at how you can apply CASP design to typical questions. So the type of questions you have can generally be broken down into 4 particular type of design solutions or strategies. And then we're going to look at how, when you want to supply your sequence to us, how you go about submitting sequence, but with annotations in order so that you can give us plenty of information about how you want your assay designed and in turn help us. And at the very end, I'm going to talk about the importance of a particular type of annotation that you can use and why it's important. And that will then move us on to part 2, where I'm going to talk to you about how you can go about screening your assays and sequences and use publicly available databases and using BLAST and simple tools online and the types of filtering options and things you can do in actually to find anchors. And what do we mean by anchors is something we'll also go through. So it's a kind of a workflow. We'll describe each one of those, and then we'll move on to actual worked examples. So I'm going to go through what I did at each stage in order to give you a nice working assay. I'll show you the before and after and particular examples of each one. Before I'm going to move on to specifically targeting regions within complex genomes. So we're going to use a couple of examples, wheat and potato, and we're going to look at the effects polyploidy can have. And they're 2 discrete examples or organisms, but the question that we're asking is different of each. So I've given you a few examples so that we can move on and give you a good overview of how you go about doing this and hopefully get some better assays and simply put, better by design. So I'm going to go through quickly who we are. We're one of the longest running, probably, production genotyping installations in the world. And in the far left-hand corner of the picture, you can kind of see the scale of the laboratory that we're working in over in Hoddesdon in the UK. And this is specifically for CASP, but we do all sorts of other things here. And what the view you're seeing there is from the extractions lab. Then assay manufacturing is in the central image. Everything's processed and part of a project. All the oligos are received and we manufacture the assays on site. And then to give you an idea then just how many data points that we're dealing with, or how much throughput we're dealing with, that's the far right picture gives you the area that we're dealing with for DNA arraying, which is probably about double that. And you can see in all of those shots different mechanized solutions that we create in order to get around bottlenecks in order to speed this process up. So just on the pie chart on the left, I wanted to give you an idea of the sort of the distribution of life that passes through the lab in between sort of 2016 and 2017. And you can see probably predominantly 62% of the things coming through the lab are plant. Whereas animal, I mean by livestock, and then about 8% is human. And then the remainder is made up by vastly different species and organisms, including bacteria and yeast and protozoa, insects, and of course fish, and that could be also mollusks as well. And I wanted to give you a quick idea of sort of the types of institutions and the companies that we've been working for, and I've been lucky enough to work for on HELP, which is part of my role at LGC over the years. So let's quickly move on then to how does CASP work. So let's start off with with ways that you could order assays. So first of all, we have something called CASP by design, and that's where you submit sequence to us. In effect, you send a sequence to us, and we'll do nothing more but design it through our in silico primer design software. There isn't any other interaction with us, and then the assay is delivered to you. Whether or not it works, that's down to you in optimization. We can help, of course, but we also offer a secondary service, which is CASP on demand, and again that goes through the same a similar pipeline. And essentially prime design, but then we also will then do blast and other analyses to actually get your assay working. And what we'll also do is we'll also test this in the lab. So if it's not human, if it's human, sorry, you'll be able to supply us the assay, and we'll have DNA in house. But for any other species, it doesn't matter which one. You can provide us with DNA, and then we can test it for you. So those are kind of the two major pathways to ordering CASP assays from us. So what's in a CASP assay, and how does it work, and what are the components of what we would call a CASP reaction mix? So you can see in the far left-hand corner there, so you have CASP assay mix, and that's made of two forward primers, and we call them allele specific 1 and 2. One corresponds to FAM and has a tail sequence that corresponds to the FAM cassette, and one has a tail sequence that corresponds to the hex cassette. And then, obviously, to complete the PCR circuit, we'll have a reverse primer. The 3' end, and this is a very important part of how CASP works, is specifically tailored to which allele that we're going to address. So in the example there, we're looking at allele 1 is C, and then allele 2 is going to address A. And then in the master mix, this contains the fluorophore and quencher cassettes, i.e. the quencher is bound to the fluorophore cassette, and both of them are oligos. The quencher is in proximity and quenches any signal, so there is no background at the start. The master mix contains the enzyme, DNTPs, magnesium, everything that it needs to support PCR. And then the final part is the template DNA. And this is the part that is obviously provided by you, And that's essentially the three things that make up a CASP reaction. So I briefly mentioned why is it important for the 3' end. So if we have a look at how PCR works, essentially CASP is nothing more but PCR with an additional tail sequence. Now, when it's what we call a competitive, we have 2 primers. One is almost identical to the other, except for the 3' end, and that's the important part. So as we denature and then we reduce the temperature to annealing, the 3' end will address whether or not the SNP that we want to look at will actually enable it to bind. So here in the picture, in picture 2, here we're looking at C binding to its complement G. Now, the polymerase within CASP is completely dependent on the first few bases being completely bound and a match. Otherwise, polymerase falls off and there's no extension. Again, there's a reverse complement primer to complete the first round of PCR, but at this point there's no signal generated. Then we look at the second one, you've got round 3, or sorry, not round 3, but round 2. You're essentially completed your first amplicon, and it's the reverse primer now in the second round of PCR that's reading through and creating the anti-complement of the tail. Now, the reason for the anti-complement of the tail is so that when you add the primers and everything together in the reaction mix, you're not getting the quencher and the fluorophore binding to primers. So it's not until you get the anti-complementary sequence will the actual cassette bind in with CASP and start to generate signal. And this is what we see now in round 3 of PCR. And it's at this point where the cassette is now starting to incorporate into products. We've addressed each one of the alleles specifically, and essentially we're amplifying up. And this continues on and on and on and on and on, and you progressively get more and more signal. So for a really, really good in-depth explanation, there are links in the webinar view that you can go to have a look at how does CASP work. And there are 3 videos, and I recommend taking a look. So we've had a quick look through how CASP works, and it will become more and more evident, and as we go on, how the 3' end of primers and the way that interaction works with the polymerase, and how that codependency, in order for amplification to occur, can lead to some very nice tricks, and actually is very, very specific. So you'll probably have questions about what type of variation that you're looking at. So we can pretty much boil these down to 4 particular groups. Now, an indel, I suppose, is an indel, but we've broken them down into 4 discrete groups, purely because small insertions or deletions can differ slightly, just in the way that we, in terminology for us, but this is more a solution to the way that we design the assay, more than realistically it being any different to any other type of insertion or deletion in its truest sense. So the first one we've seen here is a single nucleotide polymorphism, and it's A or it's G, and then we design one allele specific primer to address either. So a single base insertion would be either it's there or it isn't, or it's been inserted. A single insertion or deletion would be a single base has been inserted, or a small insertion up to 25 nucleotides long has been inserted, and that's what we'll call a small single insertion rather than single. It's a fine line in distinction, I know, but actually it's something that we reference. And then large insertions or deletions. Now, this could be one base being replaced by up to 26 or bigger. Actually, and it doesn't matter however big, as long as the breakpoints are known at one end and the other, or there is sufficient sequence within the region that we can design another type of oligo solution, which I'll come on to in a minute. then it could be any site. So this could be wholesale rearrangements, large whole gene deletions. So thinking about how we go about, or what type of insertion we're looking for, or what type of variation we're looking for, can give us our solution, the way we're going to go, and how we're going to design. So with a standard CASP assay, a typical assay, as we've seen already, with our forward primers, or our allele-specific 1 and 2, one will address the A, and the other one here, I've got the 3' end, the final base, addressing G. And to complete the PCR circuit, I have a reverse primer, which essentially works with both. So the next slide gives you an idea of what a typical assay would look like. If you're familiar with genotyping, this is pretty much a plot. So just for ease, and for the rest of the remaining, I've essentially used allele 1 is blue, allele 2 is going to be red, and then obviously the heterozygous, or the essentially 1-to-1 of each, is green. So here I have a single base insertion, or a deletion, of course, and with this one here I've said that allele 1 is going to address the A. But what I have got is I've got a very similar primer now, but what I have got is a skip. Actually, what I'm addressing is the last 2 bases at the 3' end, and one is G and one is A, and then for allele 2, it's going to end in GG. Now, that's a slight difference, and it is just differing by a single base, but what I am doing is I'm skipping the insertion or deletion, and I'm looking for the next base that is unique, the next unique base that I can use to differentiate one from the other. And again, a common primer completes the circuit, and here's this assay functioning, and you can see the genotyping displayed there. So now similarly we're going on to insertion and deletion, and we're going for 2 or 26 bases. And the distinction here will become more evident a bit in a second when I explain sort of what we call megadels, or mega deletions, or mega insertions. So again, what I've done here is I'm looking for the next set of bases that are specific to the target, or the regions I'm looking for. And all I'm doing is addressing where the breakpoint starts and begins. So for our insertion here on allele 1, I'm going to put the 3' end bases are going to be A and T. And then for the allele 2, which is going to be our wild type, without the insertion, and that's going to end in T and then C. So those are the 2 bases that differentiate, and that is sufficient to actually differentiate between the 2. You'll notice now that where the common primer is, the distance internally is slightly different, okay? And that can have an effect, as with anything, as with any sort of PCR, on how well the efficiency works and how groups tend to move. So ideally you wouldn't want to go any further than we have here. We've got probably about 30 bases internally, and that's generally going to be okay, but it would be a case-to-case and design by design. And as before, just a single common primer completes our PCR circuit, and here's that assay running. Now I'll just point out on the group that you've got here, so when I ran the assay, I got one that didn't actually fall in within the group particularly there, so I've coloured it pink. So just as a familiarity, and we'll be seeing something like this a bit later on, but actually this becomes a little bit more useful information about what's going on and what potentially could be going on. So then finally we have our large insertion or deletion. Now here is an overview of what we mean. So you notice here that I've got the allele 1 primer and I've got allele 2 primer, but I've also got reverse primer 1 and reverse 2. So now I've made a 4 primer circuit. So essentially I'm requiring 4 primers. And all I'm doing again is, as the same with all the other examples, I'm addressing the basis or the sequence that I know. I'm looking for where that breakpoint is, i.e. where the new sequence has been inserted and what is that change from, or where that change is going to be from where that sequence is removed. So you can think of it where you slice down the middle, and the sequence for the primer, in the most part, for allele 1 and allele 2 are roughly identical, and then it's the remaining bases that I'm going to, or you're going to use, in order to differentiate. What is the differentiator between the two? So what's the new sequence versus what's wild type, what's been deleted? So that's what I mean by breakpoint. I think the next issue, or the next image here, gives you a better idea of what I'm doing, or what I'm saying. Now, as I remember, I mentioned earlier that we want to keep how efficient it is as balanced as possible. So we want to keep the PCR as balanced as possible, so that we're not skewing clusters one way or another. So what I've done here with A2 wild type, I'm addressing the 3' end as T, C, T, G. And then our large insertion. Now, this could be, as I suggested, could be any length of base. We tend to say 26 bases and up, but this could be any length of base. So you wouldn't necessarily even need to know all of the sequence within this region. As long as you knew maybe 20 or 40 bases within one side, that should be sufficient in order for us to place a common primer, and also then know the breakpoint. What is the difference, or what are the two different changes? So here for the A1 primer, I'm addressing A, T, T, A. So I'm using 4 bases as a differentiator, and that creates instability, and the instability is also very good for specificity. So here you can see that I've run that assay, and again you've got some pink that haven't amplified in this instance. That may be indicative of something else going on, but you see 3 clusters as you would expect, and it's exactly the sequence that I've shown you here is the design that we're running. So let's suppose now that you've had a look at the sequences and you've got an idea of what it is, the type of mutation variant that you're looking for to design. And again, so if you were looking for insertions and deletions, that kind of thing, it could be that you're looking for CRISPR mutations. You perform some kind of mutation. You've seen wholesale deletions, something like this. But you know probably more about the sequence than we will. I mean, we'll take these sequences in, but there is a way that you can direct design. So from the knowledge that you have about the sequence and what's doing and what's important, it could be, for example, that you have a knowledge of varietal information that's important for placing primers. So armed with this, you can give us that kind of information. So how would you submit a sequence? So the first one, or the top example one, it's essentially A and then \T, or whatever your mutation is, for example, A or G, so on, within square brackets. Simple. You can also just apply square bracketed and then the RUPAC code. You can provide us with RSID numbers. So here we've got 1, 2, 3, 4, 5, 6, and then we will download that sequence and run and submit for design. Indels are slightly different, where that if you're looking for an insertion or deletion, that we would expect that there'd be the sequence that you're looking at is wild type, and then you would / and then T is either the inserted base or it's the deleted base. It could be any number of sequence or a length of sequence. For example, if you were looking for a whole gene that had been deleted, it would be sensible probably to provide us 100 BP, 5', 3' in, flank with N in the centre, and then flank those N with another 100 BP, which then is finishing off 3' to then out into the wild type sequence again. So then we'll understand what it is you mean and then design accordingly. So there are some ways that you can mark special requirements, and effectively what this does is it's telling our automated software what it is you want to do, and certain things have a particular meaning. So for avoiding bases completely, you can place n, and n's we will avoid completely. But what you're also doing, you have to be quite aware of here that for the example I've got here, where I've placed nn next door to my SNP, it will completely avoid that direction. So it will only design the common in the reverse direction, the allele specific in the reverse direction, with a common primer in running forwards. So you've dictated in which direction that the primer should be designed. So there's two things there. You can say, I believe, or I'm unsure of the sequence, just flanking 5' here, or I want to avoid allele primers going in this direction. So moving on then, slightly less, if you like, stringent is suspicious bases. So you can inform us of sequence that you're not completely sure about. It's slightly ambiguous. Maybe you have sequence of results and it wasn't completely clear. Again, what we'll do, it will shift design in one direction, as the first example has shown. But if that isn't possible, then what it will do is it will use that as a last resort. And then, obviously, if you've included those kind of things, we will contact you to say, are you sure we can try this? Then, obviously, you've got the situation where there are surrounding SNPs, and that happens a lot. So from different varieties or different mutations in human have different minor allele frequencies. So within a population, you might find that actually it's pertinent to say these are quite often in linkage with the one I'm looking at, and then you can put the ambiguity code in. So what we will do is we can design primers freely over probably about 2 for the reverse primer or the allele specific primers, and incorporate that into them, and essentially that's okay. More than 2 becomes quite tricky because that's the amount of species or the amount of primers, duplicates that you're making. So synthesis becomes a little bit trickier for those. Also, if you're going to say have trinucleotides, we will completely avoid it. It will treat that as an N, so it will completely avoid that region altogether. And then one final marker is the 2 chevrons bracketing a base. And this is what we say is what is an anchor. So what is an anchor is effectively a region of sequence, or it could be a single base, as I've indicated here, with two chevrons surrounding the base T there. And that's the single base that's unique and is unique to the sequence, the target sequence that you're looking at. And it's quite important because as we've mentioned before, the 3' end and the way that the polymerase interacts with the 3' end of our primers, the specific primers, is what's driving specificity to detect the SNPs that you want to look at. So conversely, you would say the 3' end of the common primer can be selected in the same way. And this is true. And we're going to dedicate the rest of this talk to showing you why that is, how that works, and then lots of examples of how that actually it being performed. And it's one of the things that you could do quite easily if you follow this essentially this pathway that I'm running for you now, that you could do this kind of analysis anywhere and with freely available tools. So those are kind of special requirements for how you can give us information to help you design assays that work. Okay, so let's now try to discover what or how we can go about finding anchors. And what are the type of things that we need to do? So when we're checking our sequences, we need to blast and we need to actually screen against the databases to which we are actually addressing. So if we're going to use BLAST, we're not going to use the non-redundant data sets. We're going to use the genome specifically you intend to analyse. And what we're going to do is we're going to check for regions of identity. So could it be anchored, or more favourably, could it be avoided? And again, using the symbol that I've just shown you, actually we can bracket whole regions of sequence rather than just trying to find individual regions that are one single base, for example. So if we can avoid having to discriminate against lots of different sequences that might cause off-target, that's probably preferential. And then we're going to compare with other data sets if it's possible. So if there are multiple genomes that we could reference or we can take a look at, so we can see whether or not the unique bases are consistent. Are they consistent between different varieties, for example? Are they the variation between the gene, or are they variant dependent? So variant dependent can be useful at some point, but actually if we want to target specific genes, regardless of the varietal, then what we need to do is we need to make sure that it's consistent. And then we want to compare the variant with the alignment. So have you spotted something strange? When you look down the alignment, do you notice that actually what I see is in different regions, the component of the SNP, for example, if I was looking at G and I was looking at T, and I see very, very similar sequences throughout the genome that also have that specific position, G and T, and then am I going to get a contribution from those different places to a genotype? So is it a real SNP? I think is probably what I'm saying. So this is a good stage actually to take a look and ask some questions. So the other thing is that not all species will have the luxury of multiple databases or even a single complete genome. Some might be GBS, or they might be things like EST tag libraries, transcriptomic libraries. And it might be necessary to discover what works. And in particular, you have to remember that where the exon bridges are. So for EST tags and transcriptomic libraries, you're going to be looking at transcriptome. So you'll have to remember or have to figure out where those bridges are, because we're going to be now working in genome from genomic DNA. And so those things will actually matter. For example, is your SNP right on the bridge? Then, for example, your reverse primer is 200 more bases away from where you're actually intending to target, and then that won't work. So you don't need many tools to actually do this, and BLAST will be the most exclusively used for the rest of this talk. But there are probably Clustable is something else that you probably might want to use as well, and that will allow you to align in sequences that you have. So if you have the ability to if you've got Sanger sequences, or there are multiple submitted sequences within a database, you can use Clustable very easily to create a nice lineup and then mark and use the annotation guide that I've given you earlier to say, okay, within these sets of sequences, actually only a few of these are actually real differences. Actually, these are differences between sequences or people. These could well be surrounding SNPs. So we'll mark those as either I'm sure or this actually is a surrounding SNP. So you can mark with N, or you can mark with ambiguities, or if there are sequence regions from that you have that you're unsure of, use the lowercase. So when we're going to go and do BLAST, we'll be using BLAST N, and we'll not be using MegaBLAST or other versions of it. So we'll use BLAST N. MegaBLAST effectively looks for very similar sequences, but what we want to do is we want to look for very, very almost to the point dissimilar sequences. We want to look for anything that might produce a hit, that might sequester parameters away from our reaction and ultimately cause our assay to fail. We try not to use masks because essentially this hides problematic sequences. Repetitive regions are a good example. And essentially we can quickly create and search alignments just to check where our parameters are likely to hit and where we might be able to then avoid. But also, am I unnecessarily trying to drive, as I mentioned before, with I could avoid actually using a single base. Is there a clear sequence away? So am I able to complete an amplicon circuit with a set of primers? Could I actually move a little bit further out, place my common primer, where now I don't have to anchor, now I'm actually within the target sequence. And so when we look for highly conserved sequences, these can potentially give the wrong or very misleading results. And I've got an example for you coming up in a minute. So when we land on the blast page, we're presented with some options. So we're going to use nucleotide blast, but what we're going to do is we're actually going to select the genome that we're going to analyse, the organism that we're interested in. Okay, and in the database search, we can type in, if you have the taxid ID and you know that, the common name or the scientific name, you can place that in there and search. Okay, warning that not everything is here, or not everything is up to date. So if you're working on things other than sort of model organisms, for example, hops, very good hop base, has very up to date sequences, same as Solcat for tomato, up to date tomato databases as well. And what we're going to do then was we'll paste our sequence into the paste window. So we've clicked on and we've searched, and we've got a database window to come up, our blast search window, which will paste our sequence in, but we'll remove the square brackets. And if we've chosen to use base with a / and base, we'll replace that with an ambiguity, so that we know where we're looking for. When we do the alignment, we know where our SNP is. It makes actually finding out or finding where our SNP is in relation to the rest of the sequence much easier. And then what we're going to do is we're going to click on the algorithm parameters tab at the bottom, and we're going to make some adjustments. So as I mentioned to you before, we want to screen for everything. And we want to make sure that we can see as much of the sequence as we can. And normally I'll reduce this down to the maximum return hits are about 50. If you've got more than that, then you've got other issues. But so 50 return sequences is best, and we don't want to tie up NCBI's time too much. And then I'm going to switch off the filter, both of those, and I'm going to switch off the masks. And then I'm going to leave everything else the same. That's the only adjustments realistically will work 99% of the time. If you just want to do a quick screen, obviously, more complex situations might require, will require more complex situations. But this is a very good, quick and easy screening tool to allow you to do some analysis and guide prime design. So once we've done our alignment, we run it through NCBI. And we've got a return result, we're going to reformat. So we can view the query in what we call an anchored alignment. And this just makes looking across the sequence very quickly much, much easier. So remember, all we're going to do is we're going to leave the HTML part alone. And then the second line down, the second option for the tab, we're going to change to query anchor for alignment. Okay, and then what I tend to do is where the base length, or essentially, how many bases will it run before it will overwrap, or before it wrap to the next line, I increase that to 150, and it just makes viewing it very quickly much easier. So here we have an alignment view. So you'll probably be quite familiar with this, but this is that where we've adjusted the actual view on the output through NCBI. So you can see that where I've marked Y, and we've got T and we've got C underneath our Y in our alignment. And then we can see the top hit is the sequence and target that we're interested in, and then we can see what other sequences are around and potentially might interfere with our assay. So this way, what I've done is I've decided, well, I'm going to design the allele-specific primer in the forward direction, because what I can see is actually if I place my primers a bit further out and I bracket in this region, I can avoid any of the potential of creating a complete amplicon with the sequence in the third row down. And this sequence over here, it's okay. I'm not going to make any complete amplicons, so I don't have to find an anchor to avoid. So I've literally marked with the chevrons around a region of sequence that I consider is clear, and will design in that direction. So now we've had a quick look at just how I go about doing it, what the things that you can do to run it, what you're looking for. Let's actually look at some worked examples. So earlier on I mentioned where SNPs that are not SNPs, where they can make a contribution to the genotyping you're seeing, and actually give you misleading results. So HCAR2 is the gene that we're interested in. HCAR3 is the pseudogene. And they have two different SNPs. So on HCAR2, I'm interested in 590447, and that has a minor allele frequency of about 0.01. Whereas the higher frequency SNP for HCAR3, RS57187746, has a MAFA of about 40%. And they're about 95% identical. So what I've done is, you can probably faintly see here, is I've highlighted two differences. And so what I've done is I've designed the allele-specific primers to run in the reverse, and I'm going to switch between two different parts of the genome using just a single base. So for our HCAR2 example, I'm going to use just the 3' end of the reverse primer, and I'm going to address G. And for HCAR3, I'm going to address the 3' end with T. Now that is essentially the only difference between the two assays. And what we're going to have a look at now is, what difference does that make? So if I don't anchor it, if I just place the common primer anywhere, and I don't address the 3' end to a specific base, I get a genotyping assay that looks like it could be okay. What I'm noticing is that the allele 2 group is pulling in quite significantly away from the y-axis and into the sort of the central region, the heterozygote group. Now, that may or may not be right, but let's have a look at the central example. And actually, when I anchor just to T, I notice that the frequencies have changed. But what you also get is that now that the groups are now falling to the correct points in the axes, the red group is against the Y and the blue against the X. So what happens then if I address the G, which is specific to the one that we're after, RS590447? Well, what you can see is that actually the frequency is completely different, that I am actually targeting one specific region over the other. So I can switch between two different points on the genome by using a single base. Okay, so let's look at another example. Here is an example, RS13068501. Now, you may look at the sequence and go, well, why have you gone this way round? Why did you not go the other? But actually placing the allele-specific primer in the reverse region actually addresses quite a lot of low complexity sequence, and that version didn't work at the assay design. So that leaves us no real choice then to place a common primer in there without lots of hairpins and other things. So we have to move out to the more complex sequence, but that brings about its own problem. That's brought about very similar sequences across the genome, but again, what I've done for you here is identified a single base, and in this time I'm going to use G, and I've highlighted it around the chevrons, and it differentiates G from all the others here is A, RA. So here's the assay before unanchored, so you can see that I'm getting a contribution of multiple different regions. It's not working terribly well, and I'm just getting a kind of a group of non-specific amplification in the center. Now, here it is anchored. Now, remember that I mentioned that this is PCR. So a lot of the things that you can do, optimization, temperature, as well as additives, can be used to improve CASP. So in the central one, I've used our standard conditions 6155, and that's a touchdown PCR. And this is anchored. it looks like it wants to do something else, or it wants to be doing something, but it's not great. But noticing that we're essentially anchoring to G, and quite the primer itself is quite high GC, that the annealing temperature is slightly higher, so actually increasing the temperature to 68 to 62 solves this initially, but not brilliantly. But then I've added additional 5% DMSO, and then you can see that the result on the far right. So much cleaner genotyping, and an assay that scores and corroborates well with the expected frequency we tended to see from NCBI. So now we've had an idea of, very simple diploid genomes, quite simple examples of how we go about using an anchor, and that actually we can selectively choose between different regions within a genome. We can actually employ this now into more complex genomes. So one of the very good example here, and this is probably a simpler example, we like to detect a SNP on 2bs of the wheat genome. And again, I've highlighted for you that I'm going to select the base A in this example, and I've got two choices, either 3' or 5' to the SNP or after. One point of note here that essentially wheat is inbred, it's hexaploid, there are 21 chromosomes, but essentially it's 3 organisms in one, and essentially they are labelled A, B and D, and they make a different contribution, and oftentimes it's required or to know what allele or what part of the genome structure is actually contributing to a phenotype, for example. So we can use the same methodology. We'll screen again, and we want to come away now, and we want to target 2bs only. There's a duplicate copy that's very similar to 1bs and 2ds. And you can see there are four alignments there. And again, here is the example on the left of the anchored or unanchored, and then the anchored example where we've chosen A, base A, and we've anchored as well. This is standard conditions. There was no other optimization other than just select which is the unique base and then design and run. So that's quite a simple example, very similar to what we've kind of just looked at with the diploids. Here is a more complex example. Again, we've got more options in this instance. And what we can do, we can see, again, the unanchored example doesn't give us very good genotyping. We can see that there probably should be 3 groups here. But again, with the anchored example, we get nice 3 clusters. So our final example is where we're going to look at potatoes very briefly. And again, they are polyploid, but they are tetraploid, and they differ from wheat in that we're not often trying to screen for different genomes, or that's perfectly possible to do if we know what the anchor is that's specific to the genome contribution. But we're employing the things that we've learned earlier on, where different parts of the genome or different SNPs can have a contribution or an effect on the data that we see. And in this instance, with potatoes, they're alloploidy. So what you'll find is that you'll have a contribution in terms of how much or how many copies. So not just 2 genotypes and a heterozygous example, but now you can have 2 homozygous groups, a heterozygous group, and because we're now working in tetraploid, we have the option of a middling group between both homozygous and heterozygous allele 1 and a middling group between allele 2 and the heterozygous group. So the potential of 5 different groups. So interestingly, can you actually design CASP assays to that? So again, we're going to identify the anchors, And just to give you an example, what I'm going to do is I'm going to show you unanchored. And I've selected the anchors that will target the particular region that we're interested in. And that's the one we want to measure an allele dosage. How much is it contributing? So is it 1T or is it 4 Ts and an A, for example, in this example? And then what I've done is also then the most similar sequence, I'm going to show what the contribution of that might be. So the effects of not anchoring, the effects of anchoring away, which are the chevrons using that I've coloured blue, and then I'm going to select the A, which will target our specific genome, and then what are the effects that happen there. So you can see with the unanchored example, you can see that actually you're getting a kind of a spread of groups, but then you're getting quite a large pull towards our Y allele, or hex group, if you like. And you can see that we're not able to score very well, but it wants to kind of separate into multiple groups. It looks like it should, perhaps. So then for a middle example, I've anchored to G, so I've anchored away onto chromosome 6. And that sequence we saw just now, that's very similar, but you can see that it has its own contribution. But again, I'm getting some that aren't actually amplifying. And in this instance, it was effectively, it's detecting a SNP within a SNP. So at that point on chromosome C, the minor allele frequency is for an unknown SNP. There is another SNP there that isn't being detected. And that's why they're not amplifying. And then finally, when we correctly anchor for our target region on chromosome 2, we can see quite simply there are 5 groups, and each one of the different samples has a different contribution of each one of the alleles. And as I explained, you can see that there's the AAAA genotype, AAAB genotype, or heterozygous genotype, or ABBB genotype, and our BBB genotype. So again, it's very easy to do. It doesn't work with all assays, but that's probably because for the 200 or so that we've tested, some of the varietal differences will have an effect on how primers work, how primers place. So the more you know about a population group and how you can then annotate and give us information about it, it will drive how we can design or what tricks we can employ in order to actually get this working. But this worked very nicely, and of the 250, probably about half of them work straight away. So perhaps just to summarise, using a very simple set of guides, as I've explained to you, will help you transform your data. Thinking about how sequence has changed, what type of variations are you looking at, will help you select the most appropriate design method or design strategy. Using the annotation system then, as you've identified what you'd like to design or how that system's going to work, will allow you to annotate and tell us more information about what it is that you want to do, and we can then design straight away your CASP assays. And that hopefully just our brief touching on CASP and how it works, it's a PCR technique. So things and examples I've shown you as well where I've altered thermal cycling or I've added additives can help you optimize and improve assays too. But one of the most fundamental and important part of understanding any of this part of the system is screening sequences prior to submission. Then you understand what the sequence is doing, and using just simple blast alignments is often enough. Although, when you're selecting bases, use more than one. If you're unsure, you can identify multiple different ones, and then we'll try and screen between them. So identify it as much as you can, but obviously if you can avoid having to anchor and find regions of sequence that are unique to your target, that's always best. And if you, for example, the workflow is probably all right for maybe 100 or so sequences, but if you have more or you don't feel comfortable, or you would like someone to wet validate it, then you can always use our CASP on-demand service. This will allow you to do large numbers of assays, and they are also wet validated in our laboratory. And again, any organism can be sent to us as long as you have DNA. It works on anything. DNA is DNA. So thanks very much for attending. I hope it's been informative. And if you have any questions, I can answer those now.
Thank you, Dr. Curry, for your presentation. A quick reminder for our audience on how to submit questions. Simply type them into the drop-down box located on the far left of your presentation window, labeled Ask a Question, and click on the Send button. Dr. Curry will answer as many questions as time permits. The first question is, do you offer this type of service for all CASP assays?
Yes, so any organism, as I mentioned earlier in the slides, there's about 402 organisms that we encountered over about 16 years. We have in-house human DNA that we can run your assays on, but if you have variants which are quite rare, you can supply us controls, or for any other organism, you can supply us DNA, and then we'll design and work with you to go get hand in hand to get those going. It's important to note we'll also supply you the data as we're working through different iterations of designs, and we're running in the lab, so that you get a good feeling of what's going on, and we'll work together as a kind of a project.
Thank you. Do you offer a design service for any organism?
Yes, any organism.
You mentioned detection of multiple groups towards the end and referenced a potato example. Was this the only example, and does this work for all organisms?
Well, this is the only organism that I personally tested myself. We know that it does work in others. Did it work for all assays? No, I screened 200, 250, and probably about 3/4 of those worked. And that's probably because we don't really understand what the variation between each one of those different germplasms that we tested is. So it could be some of the cases the primers hit a surrounding variant and wouldn't work, or we didn't know enough about them to get them working. But yes, in a large contribution of those things, they did work. And there's one other example that I know that's published by Janet Fulton, which looks at HLB in chickens, and she's looked at copy number variation and how she's detected that with CASP, which is quite a nice and interesting publication. So those are about the only ones that I know so far, but it's quite possible that it probably would work with others as well, I guess.
I would like to once again thank Dr. Curry for his presentation. Do you have any final comments?
No, but hopefully if you have any problems or any questions, please feel free to send them over to us, and we'll do our best to help you out. Thank you.
Well, thank you for that. And I'd like to thank the audience for joining us today and for their interesting questions. Questions we did not have time for today will be addressed by the speaker afterward. We would like to thank our sponsor, LGC, for underwriting today's educational webcast. This webcast can be viewed on demand through October 2018. LabRoots will alert you via email when it's available for replay. We encourage you to share that email with your colleagues who may have missed today's live event. Until next time, goodbye.